Hi, I'm Cameron Burke, and welcome to this Kaplan MCAT Science Review for Control of Gene Expression in Eukaryotes. This video covers a basic definition of transcription factors, which are the proteins that work with enhancers to increase gene expression. You'll also be introduced to regulation of chromatin structure, which can affect whether gene expression is increased or decreased. The main goal of this review is for you to be able to know the individual actors who make eukaryotic gene expression happen and how they work together, so keep that goal in mind as you work through the video. Ideally, by the end you'll be able to answer questions like this one about decreasing gene expression, both in practice questions and on test day. Usually before gene regulation takes place, a transcription factor searches the strand of DNA for the gene whose product needs to be increased or decreased. Transcription factors are actually a group of proteins responsible for binding to DNA to initiate transcription. Each transcription factor is a large protein made up of smaller parts with special jobs. These littler specialty pieces are called domains, and there are two that you need to care about on the MCAT. The first is the DNA binding domain, which binds a transcription factor to DNA, and the second is the activation gene, which holds on to other proteins involved in gene regulation. We'll get into specifics about these domains shortly. Once a transcription factor and all its other regulatory protein friends come together to make a transcription complex, a regular amount of transcription happens, or just enough to keep your body homeostatic, and usually that's all you need. But sometimes you need more or less of a gene product to maintain homeostasis. For example, if you eat a ton of ice cream, you might suddenly need way more lactase than you typically produce. You'll need to increase gene production, so response elements come into play. Response elements are pieces of DNA that bind the specific transcription factor for the gene you need to make more products from. It works from the same promoter region that initially regulates transcription. Extra transcription factors come and bind to response elements to fulfill extra demand. Response elements and promoters are both cis regulators. In biochem, this term means that they're located in close proximity to the gene on its DNA strand. Not only are they in the close vicinity of the gene, but they're part of the DNA. The actual transcription factor is a transregulator because it comes from far away to work on the DNA. It specifically comes from another part of the cell and not from the DNA. This is totally different from the cis and trans stuff you already know and love from OChem. On test day, if DNA is involved, stick with the in DNA versus from other parts of the cell definitions of cis and trans. Response elements are the first line of attack to produce more gene products, but if you need even more than that, enhancers amp gene production way up. If that sounds similar to what response elements do, that's because enhancers are actually just a little team of many response elements who join forces for maximum impact. They don't need to be close to the gene on the DNA strand to increase production. They can work from 1,000 base pairs away, as long as the DNA folds itself up into a hairpin, so the regions are in closer spatial locations, even if on the DNA strand they're far apart. The entire transcription complex binds to the enhancer thanks to the domains that were introduced earlier. The DNA binding domain of the transcription factor docks the whole complex onto DNA, and the activation domain holds hands with all the other proteins that need to be present for transcription to work. Together, the two domains hold all the proteins together into the transcription complex. Transcription factors play a really important role in increasing gene expression, but they aren't the only ways to increase gene output. Other ways to increase gene products include duplicating a gene in series within a single chromosome, or duplicating a gene in parallel by letting DNA helicase split open only the desired gene and then having that single gene be replicated over and over. Those methods aren't super important in terms of transcription factor involvement, but they can get the job done regarding increasing gene products. Before transcription factors can get down to business, a transcription factor has to be able to get to the DNA. DNA isn't floating around the nucleus doing whatever it wants. It's usually stored as chromatin. Chromatin has to be remodeled, so transcription factors can access the DNA to do their regulatory business. There are two types of chromatin to worry about. The first is euchromatin, which is a nice, usable form, and can be identified by its light appearance. And heterochromatin, which transcription factors basically can't use without remodeling it because heterochromatin is like a DNA bank vault and appears dark when it's viewed through a microscope. In order to remodel heterochromatin to usable form, transcription factors call in helper cofactors to get heterochromatin to loosen up. Histone acetylases are one example of a cofactor that can make heterochromatin usable. Histone acetylases stick acetyl groups on the squished in impenetrable histones. The acetyl groups weaken the charge of attraction between the DNA and histones, so the DNA lets go a little bit and the histones get to spread out. Some space is created for the transcription factors to come in and get their gene regulation on. Just like always, gene expression is already happening at a typical level. Histone acetylation simply increases the amount. When enough gene expression has happened and your cells need to go back to homeostatic amounts, histone deacetylases pick up the acetyl groups, and the DNA goes back to bank vault-style heterochromatin. Histone deacetylation can also be used to decrease gene expression to normal levels by making DNA more inaccessible. Another member of the helper cofactor team responsible for changing DNA accessibility is DNA methylase. Like manipulating acetylation on histones, changing up methyl groups on DNA can either increase or decrease gene expression. DNA methylase sticks methyl groups on the DNA so the transcription complex can't bind. Adding methyl groups decreases gene expression. But taking away methyl groups, which is done by DNA demethylase, increases gene expression by creating more space for the transcription complex to bind. In that manner, it increases gene expression. Now's a chance to track your progress with the learning goals. Check out how you're doing with this question about decreasing gene expression. The correct answer is D. Methylation puts methyl groups where the transcription complex would normally bind. To recap, there are 4 main ways to increase gene expression. They include enhancers, series or parallel gene duplication, histone acetylation, and DNA demethylation. To decrease gene expression, histones can be deacetylated, and DNA can be methylated. Thanks so much for watching this Kaplan MCAT Science Review for control of gene expression in eukaryotes. And good luck when you take the MCAT!